I'm very happy to introduce our next speaker, who is Dr. Yong-Hui Jiang, who most of you know, and I don't think he needs an introduction. He happens to be one of my favorite people on the planet, and also somebody who believes deeply in this community, from everything from creating the tools, testing them from in vitro to in vivo, and actually moving that from the bench to the bedside. And so you're going to hear a lot from him and his team today with in vitro, and then the following talk is someone, Jean Alou from his team, talking about in vivo. But then tomorrow you're going to hear about the translation of that to human clinical trials. So very exciting and very happy to have him here. He's going to talk to us about what those cell lines in a dish look like for Angelman syndrome. So Yong-Hui.
Well, thank you, Allison, and good to see everyone every year this time too. I'm extremely impressed by Ryan's talk, and Allison reminded me, I'm talking to most of you are parents multiple times. Once you understand my science, then you can go to DC and request more money for the research back to the Yale. So you are much more powerful voice than we do as a scientist. Government say, okay, you always want money, but no impact. Can my slide up? Oh, okay. So already, yeah, go. That's fine. Why go back one? Oh, that's the first one. Oh, okay. So Allison gave me this title, try to go over some of the work we have done in the lab with the fast support over the last 4 or 5 years. From this morning's talk, you probably already learned animal model like mice is pretty valuable. That's what we, as many of you know me, that I created the first Angelman mouse model when I was in Dr. Bodat's lab as a student. But again, it's an additional model we created the last couple years, and it's one with larger deletion. The original one is a good one, but it's one we're going to see here later after my talk, probably is an even better mouse model. The reason we can use the mouse model is there's a lot of things between human and mice we call conserve, which is similar, particularly at a concept level or mechanistic level. However, if you go into the microscope like the ultrastructure, again, we call it DNA sequence, human and mice are different. So that's what this morning you heard a talk, mice is not human. That's a limitation using the animal model. But how do you do it in the human, particularly for Angelman? You do need a human neuron. So that's the technology common, particular, for example, if antisense oligo, many of you familiar with, was tested in the animal, it's not directly translatable to the human because human and the mice for the antisense oligo, that structure of DNA sequence is completely different. So everything you learn from the mouse model, you have to be retested, redesigned, and before you go to the human. So to overcome the limitation for animal model, we need a human neuron. The technology mentioned early in the morning, too, called iPSC, is a short abbreviation for induced pluripotent stem cell, which you clearly see the difference between human and mice. It's the human brain is 1,000 times bigger than the mouse, and even mouse is still in vivo, we call in vivo model, a live model. So since 2021, in the middle of COVID, Alice and I had a discussion, so we started this project. So yes, we need to build up a biorepository with all the different Angelman patient, different genotype. So that's what I show here. So we're happy to say we completed this project. Every individual genotype, hopefully everyone here, the family here, we all have the iPSC created at the Yale STEM Center. And we also create some of the control as for this experiment. And again, thank you a lot of you here, fast support, or individuals listed here on the last genotype. Many of you contributed. You remember, it's the middle of COVID. I had a conversation, a Zoom meeting with you, and eventually we work out how to do it and during the COVID time. So just walk us through a few key steps once we get blood from your kids and then how it was worked out at Yale. It's a pretty complicated process and a long duration too. Remember, I talked to you and go through this, we call the RRB requirement, explain to you the protocol and what we will do and at Yale, and make sure you understand everything we do. Then you say, yes, I will contribute. And then you put your signature on the paper and you mail back to us. Then we arrange into the blood collection during the COVID time. Thank you a lot for your local physician, pediatrician support. So we're able to do this. So we'll collect blood, then we will do the sort of spin down. At that time, we're going to testing a lot of things like a mycoplasma, hepatitis B, hepatitis C. And during the COVID time, we're also testing COVID for the sample we collected. Then we use the term called reprogramming. Basically, we collect a sample from blood by the special technology called reprogramming. We turn to the blood cell, we call adult cell, or the old cell, converting to the baby cell called a stem cell. So that term called reprogramming. It's a technology developed by the Japanese scientists, which got a Nobel Prize for about 15 years ago. So with that technology, then we generate using kind of reprogramming and a lot of magic process in the petri dish. Then we say, hey, indeed, we can turn to the blood cell, to the baby cell called stem cell. After that one, we make sure when we do the reprogramming, everything still maintain the good quality. So we will have to do a lot of the, we call categorization, look at the genotype, then eventually this cell was preserved. Now we can shipping out to many, many investigators in the world. So that here's the step we took, and we were going to first confirm the genotype of Angelman still maintained. And in addition, there's no other sort of genetic alteration due to the reprogramming mistake happened. It's now no longer Angelman, it's Angelman plus something. That's not good. We also did all other work together, confirm every cell is good and that's ready to go. And also we must make sure this cell, after called reprogramming, they indeed could be polypotent, which is stem cell. We use a different marker, beautiful, looking for individual markers, say, okay, this cell looks good. And also we were going to transfer the cell to the live mice, which is immune deficiency mice, make sure this cell indeed has a capability to differentiate or turn into many different types of cells, like a neuron and muscle and other, called a teratoma assay. So after that one, we keep it in the storage. Then we make many, expanding many clones and many vials. So anyone come from any institution interested, we say, okay, yeah, we have it. We're shipping to you in the next day. So eventually, and fast, Alice and the other, we work out, say, okay, this is a resource that would be good for community in the long term. We gotta make this one public. So we finally made this e-basic repository so everyone in the world, commercial entity, farmer, and investigator in the academic institution, they can free access to. So happy to say we already distributed many PI and a few companies. Happy also to say there are 2 papers using this cell line. It's under review right now. So very soon you are going to see this product in the community. So once we do this, I'm going to show you a few cases what we do, how to use this cell line to develop a treatment or testing treatment. So one thing is once you get a disease, and then you say, okay, what do we do? One thing for sure, the purpose is we need to generate a human neuron. So that's 2 technology we're using. One we call 2D neuron, basically the petri dish. You can see individual neuron in the petri dish. Second, we call the organelle. You heard this morning, we call 3D neuron sometimes, which basically it's a mini brain. It's a much more complex than 2D. It's a 3D, 3-dimensional. It's look like the human brain. Of course, it's a mini size. It's not perfect, but it's the best we can do right now. So we can use that First testing, for example, we're testing for new treatment. This one we're working on, as you know, we do this called gene editing using new technology, not viral, not nanoparticle. So we can use these cells, screen many, many different guide RNA here. We can test in 20, which you can see each guide RNA is a different efficiency. Then the pink, the highest one, tells you the best one. So that's the first thing you can do. Very easily can do in a couple weeks' time. And we also show beautiful, you can see this green signal shows you indeed, you knock out the antisense and reactivate the sense, the UB3, the green signal. It's very, very efficient. And that's another way to look at this result. Then furthermore, we create a kind of organelle, which you can see this is a three-dimensional structure. So you choose a very complex, mimicking the human brain structure with different colors, shows you different type of cell. And it also shows you most interesting part of you focus on the green signal is a UBCA. We attach to the UBCA gene with the EGFP, green fluorescent protein. So you can see after incubator, there's a green organelle or 3D neuron. You see the green signal shows you reactivation happening, editing, it's working. So with that also, you can see this particular one as we engineer with the human neuron. So we put the EGFP touch to the Angelman UB3A gene. So you treat it with the CRISP editing, you can see the reactivation happen. So when Alice and I look at this data, she said, okay, this is perfect for precision medicine. As many of the parents probably know, you're talking to each other, say, hey, my kids have a deletion, your kids have deletion, so my kids is not same as your kid. The same thing for the UB3 mutation. So that comes down to the heterogeneity or complexity, but in ideal way, you would say, okay, before you get a treatment, everyone should get a cell, and we're testing first and see whether it's efficient for this one. So this is a beautiful example. You can see this cell is a patient with larger deletion. We treat it with CRISP editing. You can see the reactivation happen. It's quite efficient. Of course, it's not 100%, but it's a pretty efficient way, as Alanson talking early, if you have a 20% versus 40% or 5-10%. So that's the future. Of course, we have no resource. We're not able to generate a cell line for every one of you, but we will use this as a showcase. Indeed, the value of this cell line would generate for future, for develop additional new treatment. With that, I'm going to summarize. Indeed, with the fast support, many of your contribution, and also particular for many family, and contributed blood, and we were able to generate 15 Angelman cell line from all different genotypes, UPD, deletion, point mutation, imprinting mutation, all covered. For that case, everyone, particular investigator, interested in study this one, so you can just send an email to me or go to eBisc. They are free to distribute for this particular, this all cell line we generated. We're free, it's a free distribution. It's one condition, Allison, very clear when we talk this project, make sure Yale allow this free distribution for everyone, including farmer, for the cell line we generated. We achieved that goal for the purpose. For sure, we show you the case that indeed it's valuable for the human neuron, different from the mice, but it's a human neuron which oftentimes eventually is needed to move into the human drug product for all different reasons. So this cell line, indeed, I think is extremely valuable for Angelman community. Oftentimes it's also valuable for other community. They can come to us as a, they call it, okay, can we have an Angelman cell line as a control? Yes, yeah, perfect. Yeah, we will give it to you to help your community for the research. For sure, this is a project we're now getting started with the without fast support, and a lot of people contributed, Allison and other from board member and all the family contribute this repository. The people in my lab, Kun Yu and Shauna, are particularly contributed for this project. With that, I'll stop here. Thank you for the opportunity.
Yong-Hui. That was great, and hopefully everyone realizes the power of your kid's blood did that, and that is how drugs are being developed, is based off of the blood of your children that have become neurons. So that's incredibly important, and thank you for everyone who committed and donated cells for this initiative that is helping all industry members ultimately.